Welcome, everybody. On behalf of Emily Balskis, Phil Holliger, Yamuna Krishna, and Jim Wells, I welcome you to this eLife sponsored webinar on chemical biology. I'm Phil Cole, a senior editor at eLife and also a professor at Harvard Medical School. And I also want to thank Cora Korzak of eLife for organizing this webinar. This represents one of a series of eLife webinars launched to provide the scientific community with a convenient way from the comfort of your office or home to hear from and interact with representative leaders in their fields. Just a word about eLife. It's an open access journal launched in 2012, sponsored by the Howard Hughes Medical Institute, Wellcome Trust, and the Max Planck organizations. It's meant to be an elite journal in the life sciences without being elitist, and it's run by active practicing scientists or scientists across all areas of fundamental and applied biology and medicine. Unique features of the journal are its relatively rapid turnaround time, reviewer consultation processes, distillation of the revision requests for articles to streamline instructions to authors, and a published decision letter and response letter. And these features have been really popular with the scientific community members that have participated. Today we will talk about research at the chemistry-biology interface and hear from 4 top investigators, each of whom has been a recent eLife author. The field of chemical biology can be broadly defined as involving the development and application of chemical tools, approaches, and concepts for the study of biological systems. Although sometimes narrowly construed as about the synthesis and application of small molecule probes, chemical biology also involves the chemistry of macromolecules, cells, and live organisms. And today we have representatives from a diverse group of scientists who will provide an overview of some of their work and their perspectives on the opportunities and challenges ahead from across the biological spectrum. So how this will work, I'm going to introduce each speaker very briefly, one at a time, and they will speak for several minutes about their work and ideas. During this time, you should feel free to submit your questions on the panel on your computer, and these will be read and responded to as time allows, with the plan to complete this in 1 hour. So the questions will be at the very end of the 4 speakers. So our first speaker is Emily Balskis. Emily obtained her PhD in the Harvard Chemistry Department, and she was a postdoctoral fellow at Harvard Medical School. She joined the Harvard faculty in 2011 and was recently promoted to professor of chemistry and chemical biology. She's been widely recognized for her work on chemical aspects of the bacterial colonizers that make up the intestinal flora, now generally referred to as the microbiome. Like at least 4 of us participating today, she is a Cambridge, England connection where she was a Churchill Scholar. So Emily, thank you for joining us, and we look forward to hearing your talk.
Thanks, Phil, and thanks to eLife for putting together this really neat webinar. So I wanted to talk about an exciting new opportunity that I see for chemical biology, and this is the study of microbial communities, also known as microbiotas or microbiomes. So these are complex collections of microorganisms, bacteria, archaea, fungi, viruses, that live essentially everywhere, including in and on the human body. So in the context of the human microbiota, most of our microbes inhabit the human gut, which is actually one of the densest microbial habitats known. And so we've studied these organisms since almost the birth of the discipline of microbiology. It's only really been within the last 10 to 20 years that advances in DNA sequencing technology have given us a better understanding of the types of organisms, the microbial diversity that's present in and on the human body. We've learned through these sequencing-based studies that each one of us has a unique community of microbes, and that changes in gut community composition, the composition of other communities, can be strongly correlated with health and disease. If we can transition the slide. Yeah, so despite all these intriguing correlations, we don't yet understand the mechanisms underlying how these organisms affect host physiology. And this is a problem not just from a fundamental understanding perspective, but also because it limits our ability to intervene therapeutically in the many diseases that are potentially linked to these organisms. And so I think to decipher how this community affects human health, we really need to think beyond gaining a descriptive understanding of these organisms and think about characterizing their functions, what they're actually doing in the body. So there's another transition here in the slides. Yeah, so we really need to think about how we can understand particularly the unique expanded metabolic capabilities of these organisms. And so then on the next slide, I've outlined a couple of the major challenges we face in understanding microbiomes that I think chemical biologists are particularly well suited to help address. And so first, we have a very poor understanding of the metabolic capabilities of gut microbes. Gut microbial metabolites, genes, and enzymes. So most gut microbial metabolic activities are of unknown biochemical and genetic origin. And there are also numerous metabolites and metabolic activities that are likely important for this community that haven't even been identified yet. And so this is a problem because it limits our ability to interpret microbiome sequencing data and get meaningful information about what these organisms are capable of doing. And so we really need better methods of making this critical link between sequencing data, genes, enzymes, and activities. So there's an animation here. My lab has been involved in thinking about this problem, and we've found that a way to sort of accelerate this link is to, if we change the slide, sorry, there's a lot of animations. Yeah, is to use our understanding of the chemistry of enzymes and metabolic pathways. So to be able to think about the chemistry of how enzymes work and use that information as we do things like mine genomes or predict the functions of uncharacterized enzymes that might be encoded in individual genomes or microbiomes. And we've investigated this in the context of things like gut microbial nutrient interactions, gut microbial drug metabolism, which was the subject of a recent eLife paper from my lab, and also gut microbial natural product biosynthesis. And so I think once you make this link between a function and genes and enzymes, that opens up a lot of opportunities to begin to accurately identify functions in microbiome sequencing data, and even start to begin to think about hypothesis-driven, more mechanistic microbiome investigations. But this then highlights a second major challenge that we face in studying microbial communities. If we could advance the slide. Okay, so we currently lack good methods and tools to study microbial functions directly in the complex native habitats in which they're relevant. So a lot of microbiome studies really rely on pretty simplified model systems. And so I think there's a real need for tools that will allow us to investigate individual functions in real microbiomes. So this could include things like, if we advance the slide, we'll see an animation that shows, we can think about doing things like inhibiting specific gut microbial metabolic activities with small molecule inhibitors. We could think about developing tools that would allow us to gain a readout of individual activities as they're occurring in these complex habitats. Maybe using these types of tools to answer questions like, where are individual metabolic activities happening in the gut microbiome? Combining these methods with multi-omics approaches could allow us to perturb a function and start to elucidate what its role is for both microbiota as well as host biology. And so I think that also this community and the idea of manipulating this community with chemical tools also could open up new strategies therapeutically. So as we learn more about gut microbiota host signaling, manipulating, modulating these types of activities with small molecules could open up new opportunities in medicine. And then finally, I'll just end by emphasizing that thinking beyond the human body, microbial communities again are everywhere, and I think that developing general chemical tools for studying these organisms could also help to improve our understanding of things like Earth's microbiomes. So I hope that this has provided just a brief introduction to what I see as being a really exciting future area within chemical biology.
Very nice. Thank you so much, Emily. So, like I said, we'll hold questions till all four speakers have gone. Our next speaker is Phil Holliger. Phil obtained both his PhD and postdoctoral work at the University of Cambridge in the United Kingdom. And I should point out that he worked with recently minted Nobel laureate Greg Winter. He's been a program leader in the Medical Research Council Lab of Molecular Biology since 2000. And his research has involved evolutionary principles to discover new polymeric materials with genetic properties. And he's coined the term XNAs, I think, and how we can use these materials to understand genetics more and to have interesting applications in biology and medicine. So, Phil.
Thank you very much, Phil, for the introduction, and for eLife for giving me the opportunity to participate in this webinar with this really distinguished faculty. It's really great honor. So I'd like to start by, obviously, nucleic acids don't need any introduction, but they're really the key, if you want, chemical component that sets biology apart from chemistry, because biology has a memory, and obviously that memory is the molecular embodiment of that memory is nucleic acids. But nucleic acids are so much more than just the molecular memory. They can, single-stranded nucleic acids can, fold up into three-dimensional structures. They can, these structures can act both as ligands and as catalysts. And obviously they can be programmed to associate in a sort of predictable way through Watson and Crick bearings. So they're great building material for nanotech, and I'm sure Yamuna will tell a lot about that. And last but not least, they're potentially a source of really powerful drugs. But in all cases, in a way, the the potential for application is kind of hindered by systemic shortcomings in nucleic acid chemistry. Now, actually, biology itself expands nucleic acid chemistry. Most notably, there's lots of epigenetic markers in the genome, and obviously in the tRNAs and the ribosomal RNAs, pervasive chemical modification to enhance function. But in terms of biotech applications, the systemic shortcomings really are that nucleic acid drugs by themselves are not biostable, because the body, even in some privileged kind of physiological compartments, there's plenty of nucleases, and DNA and especially RNA basically don't stand a chance. They get degraded very, very rapidly. Another design fault, if you want to call it like that, is the polyanionic backbone, because although it makes the molecules nice and soluble, this really accelerates renal clearance and makes it difficult to reach particular targets in the body. It's particularly difficult to get into cells, and also they have rather the four bases are really all rather similar chemical heterocycles, which have a rather limited chemical diversity. And really a case in point is that the recently approved nucleic acid drugs, I mean, they're all based on non-natural chemistries, and And this is really the sort of beginning, I believe, of a sort of golden age, I think, of nucleic acid drugs, which are finally beginning to reach the clinic. And, the rules that have to be learned kind of to make these molecules work in the body are, I think, beginning to be figured out. And this is all based on really the last 30 years of advances in solid phase chemistry of nucleic acid synthesis, initiated by the great work of Marvin Carruthers, kind of like with phosphoramidite chemistry, and this is really built on all that. But really more recently, the field sort of has moved towards kind of beginning to encode this type of chemical variation and enable enzymatic synthesis. And the big prize there is evolution. Can we move to the next slide? So, and this, the big prize there is really to gain control and expand the sort of central dogma towards, eventually life forms which have an expanded chemistry. And one of the things, that is the most notable advances recently is this sort of introduction of an expanded genetic alphabet that's work from Romsburg and also from the Benner and Hirao labs to really expand the genetic alphabet and therefore generate whole new codons for genetic code expansion, allowing you to ultimately make proteins with encoded post-translational modifications or entirely new types of amino acids. And clearly this is sort of where chemical biology crosses over also into synthetic biology. Clearly new codons could also be freed by code compression, by freeing up redundant codons and using those for genetic code expansion, but we'll leave that to the side for the moment. And really our contribution to this field here was to introduce a whole sort of family of unnatural nucleic acids, which we called xenonucleic acids. And the enabling tool here is polymerase and reverse transcriptase engineering, which then allows you to synthesize these new chemistries encoded by a DNA template and reverse transcribe them back to DNA. And this sort of gives you a way to replicate these XNAs via DNA intermediates, a little bit like retroviruses replicate via an RNA intermediate. And replication enables evolution and opens these sequence spaces, these new XNA sequence spaces for exploration. And within that, you can find new ligands, XNA aptamers, new catalysts, XNA enzymes. And you can also, we've shown this in a sort of limited way, program them to associate into simple nanostructures. And I think the idea there is that by freeing ourselves from the limitations of DNA and RNA chemistry, but retaining this sort of great unity of genotype and phenotype within the same molecule that nucleic acids give you, we can begin to synthesize, replicate, and evolve a whole host of new type of encoded polymers and unlock their new chemical properties. For example, many of these XNAs are completely orthogonal to the cellular nucleotide metabolism, so they can't be degraded by nucleases. Some of them are also incredibly stable to chemical insults like acid or base, which degrade nucleic acids. And maybe as a sort of final point, I'd like to mention that this ultimately also, this whole new chemical machinery of an orthogonal central dogma feeds into a concept of building up a sort of entirely synthetic cells, either from the ground up or sort of simplify systems from the top down. And in this area, we've also done some work, kind of particularly in terms of the bottom-up approach in trying to enable, for example, an RNA to replicate itself, but that's a work in progress.
Oh, really exciting stuff. Thank you, Phil. So we're not going to take questions yet, but please submit your questions online, and we're accumulating some, which is good, and we'll be reading those after all four speakers. So our next speaker is Yamuna Krishna. She obtained her PhD degree from the Indian Institute of Science in Bangalore, and then she pursued postdoctoral work at Cambridge in the United Kingdom. She was a faculty member at the National Center for Biological Sciences in India for about 9 years prior to moving to the University of Chicago in 2014, where she's professor of chemistry. And Yamuna has been pursuing really frontier work in using DNA as a material for imaging and packaging and delivery, and has developed really powerful sensors that are taking hold in biology. So Yamuna, thanks for being with us.
Thank you, Phil. Thank you for that generous introduction. So good morning, good afternoon, or good evening, wherever you're listening from. And you know, I just wanted to keep this slide conceptual and not get into a specific area, mine. But see, and I wanted to also talk about where eLife is going in terms of chemical biology, and together to try and see if we can develop this area for eLife, because I think chemistry offers very, very powerful tools. I'm going to start with sort of paraphrasing Feynman, with apologies to Feynman and also apologies to the biologists, where he said, you know, if you give biologists better tools, they will ask better questions. And at some point when I read this line, this was what excited me. And I realized also that really a lot of biology is driven by visualization. And I think tools that can help really visualize what is going on inside the cell can really change the nature of questions. And so then I became sort of very technology focused to find out, you know, what are the kinds of chemical signals does the cell develop, has the cell developed to really be able to set off signaling so precisely. I think I also want to hark back to something that Phil said, which is that, you know, chemical biology really has matured. In going on from, you know, the simplistic view of chemicals for biology. And that's really seen in how green fluorescence protein really changed the way we started looking at cells and cell signaling. And from there on, you know, Jim Rothman's work, where he converted GFP to pH sensitive GFP. And then after that, the development of calcium sensitive GFPs. These are all sort of reagents and probes that helped us look at how chemical signals propagate inside cells. And these have really led to very, very deep insights. And so I see really that there is scope for even greater visualization of small molecules, large molecules, intermediate molecules driving cell signaling. And so I just put up a selection of reporter technologies that I'm excited by these days. So one, of course, are fluorescent reporter technologies that are genetically encodable. And then we have small molecules that are chemically responsive. Very recently, I think quantum dots and even being allow us to look at molecules inside cells over long durations and understand the behavior of molecules in proteins and compartments over long durations to look at long duration signaling, I think is important. Super resolution imaging, as we all know, really requires specific dyes with certain properties. That is also an area that is really developing very well. And I feel that all of these technologies are really complemented by new ways to look at electron micrographs and as EM contrast agents that are genetically encodable, that are either small molecules or programmed inorganic materials to achieve super resolution in electron microscopy. One exciting area that I think we can surely develop is really specific tools that will enable correlative microscopy, both light as well as electron microscopy. I think that's an area which I think is really exciting. And in all of this, I think synthetic biology looms large. We have the capacity to create artificial reaction networks. I think toolbox to expand the toolbox of orthogonal tools, as well as designer architectures, all enable, I think, these applications. And so with this in mind, I just thought that I would sort of mention a couple of things related to eLife. I think they have a section called the Tools and Resources section that is really underutilized. And I feel a lot of chemical biology will actually fit beautifully into that. Where really the significance requirements for this are really about, is the technology really impactful? And is there a good and powerful validation of this? I mean, the biology doesn't need to have earth-shattering consequences, but really focused on a very impactful technology. And I think that is really wide open for a whole range of chemical biology to fit in there. And I also wanted to sort of tell you a little bit about what my experience has been with eLife, because we only used to publish in the 2-word journals. But last year, we published a paper, our first paper in eLife, on the use of a chloride sensor and made a measure of chloride for the first time in a live organism. Although our work had been out for about 5 years prior to that, it was only after the paper was published in eLife that we started getting hearing from the biologists, physiologists, people who work on ion channels. And today we have 8 collaborations, with one developing every month because someone new has read our paper in eLife. And then gone back to see, oh, they've actually been publishing all this work. And so it's really more than ever, I think now, it's really about getting your work out to a discerning audience and to reach the right audience. And in that sense, I feel that eLife really has great potential in that regard. And so with that, I'm going to stop and hand over back to Phil.
Yamuna, thank you very much for that great overview and for the, I think, effective pitch for eLife. So next and last but not least is Jim Wells. Jim obtained his PhD from Washington State University and did postdoctoral work at Stanford. After a highly productive career at Genentech and Sunesis, Jim moved to University of California at San Francisco in 2005, where he's professor and head of pharmaceutical chemistry there. Jim has been at the forefront of protein science throughout his career, and both fundamental and sort of applied work, including really important strides towards therapeutics. And Jim.
Sure. Bill, thank you very much. And thanks to eLife for putting this on. And it's great to hear the panelists here as well. As you can see, it's a very sunny day in California. So my face will probably be striped in sun at various points. But I wanted to tell you briefly about kind of what my lab is up to, has been for about the last 5 years in particular. And our lab is interested in how the cell surface proteome, we call it the surfaceome, others do as well, how that collection of proteins, some 4,000, encoded by 4,000 genes, changes in health and disease. And in particular, we develop technologies for pulling out these membrane proteins in an unbiased way, determining how they change by mass spectrometry first, and then by antibodies that we build, make a whole host of different recombinant antibodies for these proteins, understand then how this group changes as a collection, as a team in health and disease, and in particular we're focused on cancer. The surfaceome, as you know, I'm sure you know, is a key proteome for modulating how nutrients flow in and out of the cell, hormones and receptors interact, how the cell communicates with the outside world, adheres to other tissues, and how it's surveyed by the immune system. So it's no surprise that most of the small molecule drugs and virtually all of the protein drugs engage this proteome. So on the left here, I'm just showing actually a shot of a figure from an eLife paper that we recently published, which describes a platform that we're using a lot to determine how the cell surface proteome changes. In the upper left there, far left, you can see two micrographs. The bottom micrograph looks like a fried egg, good for me for breakfast right now. And that's a healthy cell. If you simply put into that cell the KRAS gene, the most powerful oncogene we know of, present in about 30% of human cancers, you can see that fried egg turns into a croissant. And that in turn codes for a whole bunch of internal changes for sure, cytoskeletal changes, but also we wondered, how does the cell surfaceome change when KRAS goes into a healthy cell? So we use modern proteomics methods, some developed in Bern Walsheed's lab when he was with Rudy Aebersold, where we pull out the glycoproteins, which are uniquely glycosylated on the membrane, and compare the fried egg to the croissant and ask what's different. We find things that change. We showed that there are about 60 proteins that undergo significant upregulation on the surface when KRAS goes into them, and a bunch of proteins that are downregulated as well. We take the single pass receptors there, clone the ectodomains of that using Orpheome and G-blocks and things like that, and create the extracellular domains as FC fusions shown on the far right diagram there. That is then highly amenable to selection by phage display for antibodies that will bind to the ectodomain. That construction has on it bells and whistles that allow it, those little tags on the end or on the C terminus would be biotin, allows it to be very amenable to magnetic bead sorting and whatnot. And because phage display, all of that diversity, antibody, phage, can be contained in a library of 10 to the 10th in a volume of about 50 microliters liquid. It can be handled by a robot, which we built over the last few years. We then put all of these antigens through our robot, validate the antibodies in various ways, affinity, sequence, biophysical characterization, show that they work on this bind to the antigen when it's expressed on the surface of cells. That's that middle panel down on the lower side. Once we get there, we then have a collection of antibodies that we then go through, and now using these antibodies much more sensitive than mass spec, we can then start looking at, oh, did these antigens that we found in this model isogenic cell line that we started with, do they reflect antigens that are actually present in KRAS tumors? And we're finding that, which we're very excited about. So in the end, we hope that through this work, we'll find new tumor-specific antigens that can be targeted with antibodies, that in cancer, but we're also doing this in a number of other different disease settings, like in diabetes, in senescence, things like that, looking for how cell surface proteins are changing on the surface, building antibodies to them, using those as detectors, and hopefully, we hope at some point, therapeutics. The second slide then shows another embodiment of this technology, which is to not only use mass spec to find out how proteins are changing on the surface, but can we use DNA barcode-coded antibodies for this? As you know, NGS, next-gen sequencing, has been critical in genomics and transcriptomics, but poor proteins sit on the sideline. We can't use NGS for them. We would love to, given the fact that you can multiplex, it's super sensitive, etc. And what we realized in this first panel here was the very phage that we identify through our selections for specific proteins are indeed in themselves DNA barcoded antibodies. hiding in plain sight was a DNA barcoded antibody that each of our Fab phage has. So we take a panel of Fab phage that we've identified for specific proteins. We then use those as probes on cells. We then, at the end, wash the cells to release any antibodies that were not bound. And then we take those that were and determine the signal strength of the different antibodies. And that's exactly proportional to the receptor numbers of those targets on cells. So we've used that in this first iteration to take 50 different protein targets with 200 antibodies to those targets and characterize how those receptors change when a patient develops resistance, for instance, to ALL. We see very significant changes in certain receptors and not others, which suggest new biomarkers and potential therapeutic targets. Can you reveal the next slide? And while this is really great using Fab phage for this process because they're genetically encoded, there's some real advantages to using our antibodies directly as DNA barcoded species. So we've been developing, using chemical biology methods, new site-specific chemical conjugation methods for antibodies. Phil will know about this one, the work of Amy Weeks, recently published in Nature Chemical Biology, where we developed new peptide ligases that can put antibodies and or DNA and optical probes and biotin on the N-terminus specifically of antibodies. So we're using these technologies to build also direct Fab DNA barcoded antibodies, and hope to be publishing some of that work soon, perhaps eLife, to show this, these technologies can actually be utilized and important for understanding how the cell surfaceome changes in health and disease. So that's all I'll say now. I'm happy to take any questions.
Thank you. Thanks to all the panelists. I'm going to now start to read some of the questions that have been coming in. The first question I think I know the answer to, but I will ask it. This one is to Dr. Balskis. When the human genome is sequenced, does that also include a sequencing of the gut microbiome, or is microbiome sequencing an add-on?
When you sequence the human genome, typically you may get some sequence information from microbes that might have been associated with or contaminating your sample, but typically computationally those bacterial reads are removed. And so likewise, when people sequence the human microbiome, typically there's a step where you sort of remove reads from the human genome, and for how much human derived DNA is contaminating your sample depends a lot on the body site. With a place like the gut where microbes are more dense, it's less of a problem. But if you're thinking about something like the skin microbiome, it's a very, very low density community. So human reads are more of a problem.
Great, thank you. The next question is for Phil Holligan. Could you give us a few examples of nucleobases in some biotech products?
Well, nucleic acids, presumably. Well, I mean, biotech products, they're the recently approved drugs. I mean, their trade names are nusinersen and etplirsen, and they're targeting. So these are entirely made from unnatural nucleic acids. I mean, they're short oligonucleotides. Just the example of nusinersen, it targets the splicing of spinal muscular atrophy gene, and it makes the oligonucleotide binds to the RNA and makes the spliceosome skip to the exon, which contains, I believe, a nonsense codon, and that produces a truncated protein which is partially functional and alleviates some of the symptoms. And that particular oligo is entirely made from methoxyethyl nucleic acids. These are not occurring in nature, and I think it also has phosphorothioate linkages. And both of these chemical modifications are entirely needed to give the drug its maximum potency.
Great, thank you. The next question is to Dr. Krishna. Do the DNA sensors of the sort that you develop get recognized as foreign DNA by things like the CGAS system? And if so, how do you address that?
Yeah, that's a great question. So as you know, our body is capable, our immune cells are capable of recognizing self from non-self. And the sensors that we use, and we found out this only later, actually mimic self DNA. So self DNA has a pathway where when an immune cell takes it up, it basically has to take it and degrade it and not present it on the surface for antigen recognition. And so that's a simple pathway where it goes, it's called the endolysosomal pathway. So basically it goes to the early endosome, the late endosome, the lysosome, and after about 8 hours, it's degraded. If you actually want to induce an immune response, and this is work that's right now being done in our lab, hopefully it's ready and will come out somewhere. You have to explicitly introduce immunogenic motifs. Then you will have an immune response. And so that was quite a revelation for us because yeah, we'd been trying to induce an immune response. We didn't know what we had to do. Yeah.
Great, thank you. Next question is for Jim Wells. Jim, is the antibody generation technology that your lab has established applicable to membrane proteins with no exodomains, such as class A G protein coupled receptors?
Yeah, that's a great question. That class in any membrane embedded class of proteins, SLCs too, are more challenging because they're difficult to express as soluble proteins that can be put on beads and things like that. But there are cell selections that we and others use where you can, by differential selection, get at those. They're more challenging, and in order to, obviously, because on a cell you have many other proteins, so you have to basically have a cell without the antigen on it in selecting for that with the antigen. And so there's various rounds of selection that you have to go to, and in further characterization, more that you have to do at the back end. The other challenge is that some of the GPCRs, for instance, don't have a lot of protein real estate out there. They have little, small little loops and things like that that don't often provide that much antigenic surface to bind to. So that's a consideration as well.
Thanks. Next question is for Emily. Have receptors been identified for the metabolites released by our microbiome, or is that a work in progress?
That's a great question. It's very much a work in progress. I would say that for certain microbial metabolites, butyrate is a good example. So butyrate is the end product of gut microbial fermentation of complex polysaccharides. And we've identified things like GPCRs that specifically recognize butyrate, and more complex gut microbial metabolites. There's specific cases where we've identified receptors like GPCRs or identified specific pathways by which the immune system recognizes microbially derived molecules. But there's such a tremendous diversity of gut microbes that we're really sort of just beginning to scratch the surface in terms of understanding the diversity of the molecules they're making, and also the diversity of different receptors and ways in which these molecules might be doing things like signaling to host cells.
Great, thank you. Next question is for Phil Holliger. So you recently published a paper in eLife related to a polymerase that could use trinucleotides, triphosphates, as a building block rather than simple individual nucleotides. And the question is, how do the kinetics and fidelity of that system compare with enzymes or RNA enzymes that use single nucleotide building blocks?
Yes, that's a good question. So it's actually partially explained in the paper, but I'm happy to go through it again. I mean, it's so the reason we moved to trinucleotides is because the conventional polymerase ribozymes had a great difficulty reading through structured RNA templates. So you end up with a kind of a paradox where the ribozyme needs to fold up to have a function, but then once it's folded up, it can't replicate its own template because it's now structured. So the amazing thing about trinucleotides was that the trinucleotides in a cooperative way can bind to the template and unravel secondary structures for copying. So using trinucleotides as substrates, we now could copy through essentially any structured RNA, including hairpins with melting points close to the boiling point of water. But clearly, you're moving from 4 substrates to 64 substrates. So there is a problem of accurate recognition, and the solution. There's sort of 2 strands of molecular evidence which point to how the ribozyme solves this molecular problem. One of them is very similar to how both polymerases and actually also the decoding center of the ribosome reads out the cognate triplet, and that is because the delta G, sort of the thermodynamics of base pairing are not sufficiently discriminatory to give you good fidelity. So what both polymerases, the decoding center of the ribosome, as it turned out, ribosome, and as it turns out, our ribozyme does, is they read out the pattern of hydrogen bond donor acceptor groups in the minor groove of the pairs, and they are diagnostic for the correct base pair. And that ups the fidelity by about 2 orders of magnitude. And it turns out the triplet polymerase ribozyme, exactly like the ribosome, reads this out at 2 positions of the codon, while one of them is a wobble pair. And that gives you a fidelity. This gives you a fidelity boost, and then another boost comes from a frankly unexpected property of the triplet pools, because if you think about your substrate pool of 64 triplets, for every triplet you have its own anti-triplet, and it turns out when you look at individual triplets, the discrimination between, let's say, 2 triplets isn't actually very good. But once you have a pool where both all the triplets and their anti-triplets are present, what actually happens is the really sticky triplets, which also cause most of the mispairing, they will also, form a triplet anti-triplet pair with their anti-triplet. So they remove themselves from the substrate pool. So the substrate pool essentially normalizes itself against its kind of base pairing strength. So the weakest binding triplets are there at the highest concentration, and the strongest pairing triplets are there at the lowest concentration, and that in fact gives you another triplet boost. So altogether, we get a fidelity of about 97%, which is essentially the same as the mononucleotide polymerase ribozyme, and will be more than sufficient to replicate the ribozyme itself, or in fact something rather bigger below the sort of eigenerror threshold, which is supposedly the theoretical limit for self-replication.
Great, thank you so much. They're really helpful. Yamuna, this one is for you. So is it possible to make the DNA sensors that you're building heritable so that they can be passed on from one cell to the next?
Well, that's a zinger of a question. Currently, I can't think of a way to do this, but I think there may be possibilities. DNA is actually encodable, right? One would have to figure out a way to the kind of sensors we make. For the moment, I think it's a really out there question. We've designed our sensors to be sort of like custom probes that will give you the information that an antibody can give you. It's sort of you actually ideally want this to be label free, right? So you can take samples from whatever genetic background you have and add a DNA sensor that basically latches onto whatever is there and is able to go to a particular location and give you a chemical readout that an antibody actually cannot give you. So that was where we were going with this. Genetically encodable devices certainly are possible. I think more than DNA, I would actually think RNA space might be able to offer that. And I think Sami Jaffrey's lab is really coming up with some very exciting possibilities of RNA-based sensors, which sort of can give you the a sort of meeting point between chemical information as well as genetic encodability. I think we can still go one better and start thinking about how to make that quantitative, as well as if we can have ways by which you can localize that RNA. Then I would rather choose RNA space rather than DNA space to do this.
Good, thanks so much. So the next question is for Jim Wells. Jim, could you give us an example of a DNA barcoded antibody?
Yeah, there's examples of I mentioned the Fab phage that we've already produced, where the Fab is on the surface of the phage and functional because it binds to its antigen. And inside the phage coat is the gene that encodes that Fab. In particular, the CDRs are unique to each of the Fabs that we have. So, for example, CDRH3, which is the dominant binding determinant in the antibody, is unique for each of the Fab phages that we have. And so we can put primers on either side flanking that CDR and PCR out from the pool to find out and quantify how much of that Fab phage is bound to the cells that we probe. And then using another example, so that's the genetically encoded Fab phage DNA barcode. We've made also direct Fab DNA barcoded antibodies using subtle ligase. So what we do is we have we click on to the N-terminus of the antibody using subtle ligase. We can put a peptide on that has an azide that's highly amenable to click chemistry. And then on the other side of that, we have a benzocyclohexadiene that allows us to click on to the azide a DNA molecule. And then we can use that as a tag for that particular Fab. So each of the Fabs is clicked onto with a DNA that is unique to that Fab so that we can distinguish them by next generation sequencing. Is that clear?
Yeah, I think that covers it. This is, I guess, a general question, maybe partly to me, but to any of us could contribute. The question is, it's my impression that eLife is a biology journal. What percentage of published papers in eLife are devoted to chemical biology? Will it publish more on chemical biology? So I don't know the percentage. And I should say that in addition to me, Yamuna Krishna is also on the editorial board of eLife. And it's certainly an area that we on the editorial board would love to see more papers submitted and published in. And I think like other journals that are somewhat selective, there is a triaging process which happens, and hopefully it happens quickly if it doesn't work out. But certainly we think eLife should be a home for very high quality chemical studies related to biology. I don't know if anyone else wants to weigh in on that. I think Yamuna already sort of did before.
I have one answer. Yes, we are going to build it.
And I think that's one of the reasons why we're having this session today is to develop that outreach. This is a question that is also to all of us, and it's an age-old question for people who think about how things are worded. Chemical biology research is so diverse. Is it possible to define chemical biology with one sentence? And I thought I did in the introduction, so I won't read that again. Yeah, but does anybody else have a sentence that they like?
Meaningful chemical solutions to biological problems.
Okay. Maybe we'll do one more question, and this one will go to Emily Balskis. Let me find it. Okay. How can we address the diversity between individuals in randomized clinical studies with respect to their differences in microbiota in health and disease?
Yeah, so I mean, I think this is a really important question facing clinical microbiome research. We see this being a big problem in studies like diet studies, because what people eat is so incredibly diverse, as well as the big differences in gut microbes. So I think that one consideration to keep in mind is that most microbiome studies still are focused on the species that are present in the gut, and we know and are constantly being reminded that the phylogeny of microbes, who a microbe is not very predictive of the functions that microbe can carry out. And so I think that as we start to focus more on function, biochemical metabolic function in the context of microbiome studies, it may be that there's some evidence that suggests that there may be functional similarities between microbiomes that may be very different on the species level. So it could be that in different individuals, different microbes are contributing some of the same core functions that are necessary to have a healthy community. So as we start to learn more about functions, it may be that focusing more on the functional genes and proteins rather than thinking as much about the species diversity may help to reduce the complexity somewhat. It's a huge obstacle in microbiome research, but yeah, I think my hope is that focusing on function will help.
Well, I think that's a really great insight, and I think we've reached the end of our time. So I want to thank all the panelists for really stimulating discussion, and the questioners and all who listen, and eLife for allowing this to happen. And I wish everybody a good rest of their day, whatever time zone they're in. Thanks very much.